Okay, now we're going to be moving on to cellular respiration. This is a topic that a lot of biology students find difficult the first time they learn it because it applies a lot of biochemistry and molecular biology in a way that often isn't clear. So I'll try to slow it down a bit for this lecture compared to earlier lectures. Alright, so when I was talking about enzymes in the previous lecture, I emphasized the importance of energetics in the cell and how ATP is used to store energy, and it's really useful because then the cell can then use that energy wherever and whenever it needs to. And how exactly is ATP made? Well, the most common way is through cell respiration, which is this equation here that I've definitely showed you at least twice before in previous lectures. To reiterate, a single molecule of glucose, C6H12O6, is combined with 6 molecules of O2 to produce 6 molecules of carbon dioxide, 6 molecules of water, and energy in the form of 34 molecules of ATP. But in this process, what's the specific chemical reaction through which ATP is produced? Well, it turns out there are two types of reactions. The first is substrate phosphorylation, which is when a protein or substrate ends up with a phosphate group bound to it through some circumstances, maybe because of a prior reaction or something else. What can then happen in some cases is this phosphate can be transferred from the substrate to a molecule of ADP or adenosine diphosphate, making it adenosine triphosphate, and leaving the substrate without a phosphate group. The naming of this process really makes sense if you think about it, because it's phosphorylation, or the addition of a phosphate group, and the molecule that is phosphorylating or transferring a phosphate group is a substrate. The other type of phosphorylation is oxidative phosphorylation, and this type is really used in the later stages of cell respiration, while substrate phosphorylation is generally used in the earlier parts. It turns out that oxidative phosphorylation is actually responsible for roughly 90% of ATP production. So clearly it plays a pretty important role. The details of oxidative phosphorylation are something I'll go over in much greater detail later in this lecture, so for now it's really just important to remember that it occurs in the inner membrane of the mitochondria. So the mitochondria actually has two membranes, one on the outside separating it from the rest of the cell, and one on the inside. This space in between these two membranes is called the intermembrane space and has an extremely high concentration of protons, which are denoted by an H+, because an hydrogen atom minus the negative charges of an electron is just a proton. If you remember the laws of thermodynamics, the second law was that entropy always increases, and having a ton of one thing in the same space is really ordered or low entropy. So this means, and also the protons are positively charged, so they're naturally going to repel each other. Basically, because of entropy and these repelling charges, these protons are really going to want to get out of this intermembrane space, but they're trapped. So there's a protein that uses this fact to transport the protons inside of the inner membrane, a space called the matrix. And in the process, it actually makes ATP. This only works because the protons really want to be on the other side of this inner membrane. So when they're allowed to cross using this protein channel, it creates just enough energy to help make ATP. All right, so a lot of the process of cell respiration is getting electrons from the glucose molecule and transporting them to a final location, which is then used, and those electrons are then used to get protons into the intermembrane space from the matrix, but I'll get to the details of that later in this lesson. Unfortunately, these electrons aren't magically transported from glucose to the inner membrane as we break down the glucose. Instead, they have to be brought there by proteins called electron carriers, which you can see on the screen. Interestingly, these cells used pretty large and complex proteins to the simple task of carrying a few electrons. These are NADH and FADH2, and both of them can hold 2 electrons, and when they aren't holding electrons, they are called NAD+ and FAD, respectively. By the way, these molecules' full names are nicotinamide adenine dinucleotide and flavin adenine nucleotide, because NADH has 2 nitrogenous bases, which, if you remember, make up DNA, and FADH has 1 nitrogenous base. Also, both of these electron carriers can hold 2 electrons. That's just something to remember. Okay, so here's a quick overview of the individual steps of cell respiration. By the end of this lesson, you should know all of these and generally what they do. We'll start with glycolysis, which is where glucose is first broken down into two smaller molecules called pyruvate, which are then modified in the pyruvate oxidation step to prepare them for the citric acid cycle. And the citric acid cycle is, well, a cycle in which the modified byproduct of glycolysis is then cycled through bonding with a series of slightly larger molecules until it is essentially reduced to nothing. This is the main section where electron carriers get their electrons. After that comes oxidative phosphorylation, which is, of course, as I just went over, the process of using protons in the intermembrane space to make ATP. All right, so glycolysis, as a name, makes a lot of sense if we just break it up. Glyco means sugar, and lysis means break. So what we're really doing here is breaking down or degrading a sugar, specifically glucose. And the reaction can really be overly simplified into breaking down a sugar with 6 carbons into 2 3-carbon sugars, which are called pyruvate. Glycolysis has 2 broad stages, which can be further broken down into many smaller stages, and these broader stages are classified based on whether the energy is needed, or rather based on whether energy is needed for them to work, or whether energy is being produced. So we have the energy requiring phase, which comes first, and the energy release phase, which is second. And the overall output of glycolysis as a whole is 2 molecules of ATP and 2 molecules of NADH. All right, so this is the first half, or the energy requiring phase of glycolysis. As you can see, I've really simplified everything, so the focus is just the number of carbons in every molecule for every step of the process. So for example, we start out with the 6-carbon ring of glucose, which we know because there are 6 vertices, and end up with 2 3-carbon molecules, which we know because they both have 3 points connecting them, or which are connected by 2 lines. And we assume those 3 points are all carbons, as I went over a while ago. Anyway, in this first step of the energy requiring phase, we convert the glucose ring to linear glucose and add a phosphate group to one end in the process. And this process needs to use ATP because the phosphate needs to come from somewhere, and the energy held in the bond will be recycled at a later point in time. The resulting molecule is called glucose 6-phosphate because, well, it's basically the same as glucose, with the small difference that the 6th carbon has a phosphate group attached to it. Next, another ATP is used, and another phosphate is added to the molecules, and a slight modification is made to the atoms bonding to the carbon backbone, forming fructose 1,6-biphosphate, or rather bisphosphate, which sounds really complex, but it isn't, because it really just tells us that this is a fructose molecule with a phosphate attached to both ends, to the carbons on the 1st and 6th positions. After this, I'm going to stop using names because honestly, they don't really help much, and they aren't really necessary to memorize. Instead, I'll use some simple abbreviations. The next step in this case is to split up the fructose 1,6-bisphosphate into 2 molecules of G3P. And the important thing to remember here is that each of the G3P molecules are bound to a phosphate group, and this high energy bond will be used in the energy release phase of glycolysis. Okay, now we have a quick practice problem combining content from the last lesson and this one. This is a reaction from a slide I just showed you, in which glucose 6-phosphate is converted to fructose 1,6-bisphosphate. The question is whether this reaction is endergonic, exergonic, or neither, and I'll give you a few seconds to work through this, during which you can pause the video, get your answer, and then I'll go over the correct answer. Okay, so first of all, the answer is that this is definitely an endergonic reaction. Remember that in endergonic reactions, bonds are usually formed as energy is used up, while in exergonic reactions, bonds are broken and energy is released. Here, the reaction is very clearly endergonic because a phosphate is being added to a molecule, so a bond forms and energy must be used, not released. In fact, if you remember back one slide, this process actually uses the energy of 1 ATP, and that's where the phosphate comes in. Now it's time to move on to the energy release phase of glycolysis. It's important to remember that this entire process occurs twice, because the last step of the energy requiring phase was to split up the molecule into 2 G3Ps. The The first thing that happens is that NAD plus and phosphate are converted to NADH and a proton. Remember that this NADH isn't going to directly provide energy right now like ATP does, but will come in handy later during oxidative phosphorylation. You'll notice that this step involves the addition of a phosphate group, and that's visible here as the resulting molecule is a 3-carbon sugar with a phosphate on the first and third carbon called 1,3-BPG. The next step is pretty straightforward. One of these phosphates is removed to make a molecule of ATP, resulting in 3PG, so basically a 3-carbon molecule with a phosphate on the third carbon. The next step is also really simple. This phosphate is moved from the third to the second carbon, producing 2PG, and this is followed by the production of PEP, or phosphoenol pyruvate, which is, or rather after another water is removed. Phosphoenol pyruvate is actually a really interesting molecule that also has an extremely high energy bond, like ATP does, but in glycolysis, it's really just used as an intermediate, as the phosphate is then used to make a molecule of ATP. The end result of this entire process is this 3-carbon sugar, which we call pyruvate. And I've only been focusing on the number of carbons for every molecule throughout this process because the exact structure of the intermediates isn't really important, but pyruvates is. So this is what it looks like in the bottom right here. It's fairly simple. As you can see, there are 3 total carbons, 2 of which are double bonded to oxygen and 1 is bound to 3 hydrogens. Also, there's an OH group on the side here too. That about wraps it up for glycolysis. As I said before, it's not worth memorizing all of this. Instead, the overall equation, which is that the 6-carbon sugar glucose is broken down into 2 3-carbon sugar pyruvates. Along the way, 2 molecules of ATP are used up while 4 are produced. So overall, 2 ATP are gained and also 2 NADH are too. That's really all you're going to need to remember overall. Okay, so now we're left with 2 molecules of pyruvate, which we need to convert to something that can be used in the citric acid cycle. The way this is done is by adding what's called a cofactor, which is a molecule that's recycled, which means it is used during pyruvate oxidation, then removed later during the citric acid cycle, then reused on a new pyruvate, and so on and so forth. And along with that, the reaction also removes one of pyruvate's carbons, producing a molecule of carbon dioxide and converting NAD+ to NADH. The final resulting molecule is called acetyl-CoA and is directly inputted into the citric acid cycle. And this is the citric acid cycle. The first thing I'd like to point out is that unlike the last 2 steps in cell respiration, this one is an actual cycle, meaning that it occurs over and over and over again with each new input of a molecule of acetyl-CoA. Also, this is an image I pulled from the internet, so they really kept all the complicated names for the individual molecules. But that doesn't mean you'll actually need to learn them, and they also have all the enzyme names listed out in red too. The enzymes are something I intentionally skipped over during glycolysis, because every one of these reactions was in fact helped out by a specialized enzyme, but I didn't really bother to go over their really long and complex names. Alright, so let's get started all the way at the top where acetyl-CoA is inputted. The first thing that happens is that it'll bind to a molecule called oxaloacetate, and coenzyme A is removed while water is added, which produces citrate, the namesake of this cycle. This citrate is a 6-carbon molecule because oxaloacetate is a 4-carbon molecule and is combined with acetyl-CoA, a 2-carbon molecule. Next, a few slight modifications are made, creating 6-aconitate and eventually D-isocitrate, which are both still 6-carbon molecules. However, the next step is really important as a carbon is removed, releasing carbon dioxide and converting a molecule of NAD+ to NADH. And this results in the 5-carbon sugar alpha-ketoglutarate, and of course, it's only 5 carbons because one of them was released as a carbon dioxide molecule. Next, a similar reaction occurs as carbon dioxide is once more released, and the energy gained from doing so is converted into another molecule of NAD+ to NADH. But this time, coenzyme A is added back into the mix, forming the 4-carbon molecule succinyl-CoA, and then that CoA is then removed to convert a molecule of GDP to GTP, which is really just like ATP. Basically, ATP is adenosine triphosphate, so the nitrogenous base adenine plus a ribose sugar plus 3 phosphate groups, And GTP, or glucosamine triphosphate, is identical except that it's a guanine nitrogenous base instead of adenine. Okay, so now we have succinate, which is slightly modified to convert FAD to FADH2. The diagram I used here doesn't actually show this, but FADH2 serves as a kind of intermediate that then later modifies ubiquinone and then adds the two hydrogens to it, or converts Q to QH2. For now, you can just consider that to be FADH2 at work as an electron carrier. Anyway, the resulting product is fumarate, which water is added to, converting it to malate, and finally back to oxaloacetate, which also makes a molecule of NADH from NAD+. Once more, the key thing to note here is that this oxaloacetate can then be reused to bind with a molecule of acetyl-CoA over again. So overall in the citric acid cycle, we're inputting one molecule of acetyl-CoA, then first of all instantly getting rid of the coenzyme, then we slowly get rid of the two carbon dioxides. We also get a molecule of GTP, which remember is basically just ATP. We also get two molecules of NADH, a molecule of FADH, and it's important to remember that this is just for a single molecule of acetyl-CoA. And every glucose molecule is actually broken down into two of them. So in reality, we can double all of these production numbers. Okay, the next and final step in cell respiration is oxidative phosphorylation, which I briefly but incompletely explained earlier in the lecture. Essentially, it can be broken down into two steps. The first is called electron transport, or rather the electron transport chain, in which NADH and FADH2 that's been built up during the earlier stages of cell respiration is used to pump protons from the space inside the inner membrane, which is called the matrix, to the intermembrane space, which helps build up tension, having so many positive charges in the small, relatively constrained area. After that comes chemiosmosis, which pumps those protons back from the intermembrane space to the matrix, making ATP in the process. This is a quick drawing of the mitochondria to help you. This is a quick drawing of the mitochondria to help you better visualize this process and how it works. As you can see, there's the electron transport chain on the left here, pumping protons out, which are then being pumped back in on the right to create ATP. So this is what the electron transport chain looks like. Basically, what it does is transport electrons from NADH and FADH2 through a series of proteins, which is a reaction that makes just enough energy to pump protons to the other side of the inner membrane into the intermembrane space. However, these electrons have to go somewhere, so the final electron acceptor is oxygen, which is combined with hydrogens to make water as a byproduct, which is why there is a 6H2O in the product side of the cell respiration reaction. In terms of the actual proteins that the electrons travel through, there are 4 complexes denoted with the numerals 1 through 4, as well as ubiquinone, or Q, and cytochrome C, or Cyt C. Alright, so chemiosmosis is the process of moving the protons where they want to be, which is in the matrix, and then using that energy to make ATP from ADP plus a phosphate. And this is all done using a protein called ATP synthase because, well, it synthesizes ATP. Okay, so here's a quick practice problem. Which of the following steps in cell respiration uses ATP? Keep in mind that the question is uses ATP, not produces ATP. And there's really only one correct answer. I'll give you a few seconds to work through this. You can pause the video, you know the drill, and I'll go over the correct answer afterwards. All right, now let's take a look at the answers. Right away, glycolysis is correct because remember, there's a part in which the energy in the form of ATP is required, and even though overall glycolysis produces 2 ATP, it does also use some. The rest of these steps don't even use ATP. Okay, let's finish up with a review of the process of cell respiration overall. The first step is glycolysis, in which glucose is broken down into 2 molecules of pyruvate, producing a total of 2 ATP and 2 NADH. After that, pyruvate is modified to create acetyl-CoA and another molecule of NADH, which is then inputted into the citric acid cycle, which produces an additional molecule of GTP, a molecule of FADH2, and 3 of NADH. Finally, we have oxidative phosphorylation, which can be further broken down into the electron transport chain, which uses electrons from electron carriers to pump protons from the matrix to the intermembrane space. Then in chemiosmosis, those protons are pumped back where they would like to be into the matrix, and then the energy that's generated by doing this is used to make ATP. These are my image citations, and I think that just about wraps it up for the cell respiration lecture. I'll see you all next time.